Hello, everyone. I'm going to be talking about the COVID study, or the COVID-19 vaccine in the immunocompromised study that we have here at Pitt and UPMC. This is the core team without whom this would not be possible. And so why are we doing this? We're doing this really to help us understand how well the COVID-19 vaccines work in people who are immunocompromised, because these patients were excluded from all COVID-19 vaccine trials. And there's already been a bunch of studies published looking at vaccine responses in these patients. And without going into this in too much detail, as you can see, aside from people with HIV, really maybe about half or so of immunocompromised patients don't make antibodies after getting the COVID-19 vaccines. This leaves us with patients who are afraid and confused because the CDC is saying that it's okay to relax masking precautions, and the patients are wondering if the vaccines even work, whether they should get boosters, and they all really want to be tested. It's sort of become commonplace to get tested after you get vaccinated, despite the fact the CDC is saying not to get tested, and the FDA is also saying not to get tested by antibodies after vaccines. And the reason is because immune correlates of protection don't currently actually exist. So we don't know what to do with antibody results. But it is a research question that I think is still unanswered. And so how to solve these issues? We can restrict SARS-CoV-2 IgG testing just for studies, and we can also design a study in which patients know their test result, which is exactly what we did with COVIX. This is an example of one of the flyers that we hand out to patients and that we email. And the design of the study is that it is low touch, e-enrollment, and video consent. And the objectives are to compare SARS-CoV-2 IgGs among people who are immunocompromised versus healthy healthcare workers. And we want to look at whether antibodies are absent or present. We want to look at titers, and we want to look at SARS-CoV-2 neutralization. We're also interested in looking at the durability of IgG responses as well as T-cell responses. And by the end of this month, we're hoping to have enrolled about 1,000 plus patients with different kinds of immunocompromising conditions, including transplant, cancer, autoimmune, congenital, as well as healthy controls. So this is the design. As long as the patients have completed their COVID-19 vaccine series, they'll get a blood sample that'll be run for antibodies and some other things. And for those who are interested, we'll approach them later on to get some additional blood samples as well. And in order to do this quickly, we decided to make this an EMR embedded study, meaning that the research order actually lives right there within the UPMC EMR, and patients can actually go to one of the many UPMC hospitals across the region in order to get the research antibody blood done. But it's designed in a way, and it's built in a way that it actually all gets sent right here to Pittsburgh, where it's actually run and processed. But because it's research, it's free, but the patients also get the results sent to them via My UPMC. And so where are we? So we had a soft launch April 14th and have been revving up furiously since then with a lot of aggressive advertising across social media and our EMR. And we've had over 700 patients enrolled so far. So I'm hoping to wrap up at the end of this month / early July. And so where do we have to go from here? I think that we need to next work on deeper immunophenotyping. We need to learn about the natural history of these antibodies and how long do they last for and things like that. And I think it's really important to define immune correlates of protection. And I do wonder whether checking routine antibodies will ever become a standard of care. We'll see. I'm hoping that studies like this and others will help inform those decisions. And lastly, the question becomes what to do when someone isn't responding, and options will include things like boosters and monoclonal antibodies for pre-exposure prophylaxis. I think before this becomes standard of care, it has to be studied and evaluated. We have to follow evidence and not anecdote. And I just want to thank leadership, without whom this could not be possible. Thank you.